Hello, the topic of this video is to discuss translation. This builds on the concept of transcription that we talked about in class last week. If you remember transcription, it takes the information from the DNA, which is protected inside of the nucleus, and it copies a section of that DNA into this section of mRNA, which ends up going out into the rest of the cell. We talked about the location for mRNA. It ends up going to the ribosome. So here's our ribosome, this sort of globular thing in the background. And this is where the process of translation takes place. So if we see our mRNA section here, the mRNA comes up and it lines up with the ribosome. And if you remembered when we were talking about some of the differences between DNA and RNA, we said that one of the problems with RNA is that it's unstable. The problem that makes it unstable is that it only has that sugar phosphate backbone on one side of the molecule. The other side of the molecule is left open. And one of the things I mentioned in class was it's sort of a necessary evil because the way the ribosome reads mRNA, it has to be open on the other side. So if we're looking at this, you can see that there are these new things that are joining up with our mRNA molecule. These are referred to as tRNA. It stands for transfer RNA. These have what are referred to as anticodons, which are these sections here. It's the 3 different nucleotides that match up with the 3 nucleotides on a section of mRNA. So if this is an anticodon, this is referred to just as a codon, the side that's on mRNA. You can see that all sections of mRNA, at least in people, begin with what's called a start codon. It's AUG, and this is going to allow the ribosome to know where the protein begins, so it knows where to start building that protein. So basically the mRNA, which is a copy of the DNA, ends up coming to the ribosome. It joins the ribosome here in the front, and the tRNAs are bonded to it. As the tRNA bonds to it, it drops off one of these guys. Those are amino acids. These are the building blocks of proteins. So these are eventually strung together in order to form a long chain, which is eventually the protein that the cell then uses for all kinds of different things. These things, methionine, phenylalanine, and lysine, those are all individual amino acids. The way you know which one of those to use is this handy chart. What this chart allows you to do is turn the codon, so I'll actually sort of put this here so you can see them both. It allows you to put the codon in here and figure out which amino acid it codes for. For example, if we look at AUG, we go up to our our codon chart and we follow that one out, AUG. If we make this a little bigger, you can see that that's methionine. And that is exactly what is coded for right here. We've got methionine. So for the next one, we have UUC. So we'll do the same thing. We'll take a look at our chart. UUC is phenylalanine. Let's go back and see if the diagram's right. There we go. Got it there again. So what I'll walk you through in a second is how to go from a section of DNA to a section of mRNA to eventually having a protein using one of these codon charts. So if we're starting off with our fictitious section of DNA, we've got that written down up there at the top. Technically, this would be mRNA that we're copying it into. If you just remember our bonding rules, we've got T matching up with A matching up with U, C with G, G with C, A with U again, C's with G's, and then G with C. So now that we have our RNA, what I suggest you do is break this up into sections of 3, because the cell, or the ribosome really, reads this in 3-letter sections. So this is where you have to go to your codon chart to really figure out what these things are telling you. AUG is the first one. So if we blow this up, we'll follow it. If you remember, this is how the other one started, because all of the proteins in your body start this way. AUG stands for methionine. When you're writing these things in, the acceptable convention is just to write down the first 3 letters. So methionine would be MET. So then we've got our next one, CUU. So again, you go back to your codon chart. If we're looking at this one, CUU takes us to leucine. So we'll just abbreviate that one with LEU as we're writing that in. And then the last one, is GGC. So again, back to our codon chart. It's getting a little harder to see it now. I'm trying to put that in a spot where we can see the part that we need. So we're going GGC, which is glycine. Get that out of our way. So GLY, abbreviation for glycine. Now keep in mind, most proteins are very long and complicated. They will have far more than just 3 amino acids making them up. Later on in one of the activities in class, you will work with a realistic sized protein, and it will take a significant amount of time to go through it and transcribe it into RNA and then translate it into the actual amino acids that build up the protein. But for now, I figure if you can do a 9 nucleotide section of DNA, you could do one that was 99 nucleotides long. Just so long as you get the idea down. The key with this is understanding how to use the codon chart, and I really think this organizational step of breaking up the RNA into sections of 3, and remember this is referred to as a codon, the 3-letter section of RNA. I think that makes things very easy when it comes to then using the codon chart and figuring out which of the individual amino acids that codon is coding for. So I appreciate you guys watching. Make sure you answer the questions at the end of the video, and we'll practice a few of these in class when I see you. Thanks very much.